This is part 2 of the chapter 13-14 lecture video. We're now going to look at some different mechanisms of microbial pathogenesis. So pathogenesis is the actual mechanism the microorganism uses to cause disease. In order for the organism to be able to cause disease, they have to have virulence factors, and virulence factors are often confused with this term virulence, which is basically just the relative ability of a pathogen to infect a host. And virulence is not related to severity of the disease, just how easily the organism can cause disease. So a pathogen's goal is to enter a host through a portal of entry, and adhere to host cells, gain access to nutrients, evade the detection and removal by the immune system in order to colonize. So gaining access to nutrients and evading the immune system is needed for the organism to colonize, and then eventually leave the host. So a pathogen's goal, quote unquote, this is what they want to do. So virulence factors are needed in order to fulfill the goals. So virulence factors are traits the organism uses to cause disease. So traits that the organism has to adhere to the host cell, for example. So different adhesion factors. So bacteria and viruses have surface proteins that enable them to bind to complementary host receptors. So we talked in chapter 12 about viruses' ability to interact with the surface proteins on our cells in a specific manner with their proteins on their surfaces. Bacteria have, in some cases, pili with adhesins. So this image down here shows these pili and the adhesin proteins on the tips of the pili interacting with a host cell receptor. And like I mentioned, the attachment proteins on viruses, such as the spikes or the capsomere proteins. Capsule production also helps some bacteria to adhere to host cells. And then there's biofilm production, also allowing the bacteria to attach as well. These pili, like I mentioned, they can be specific to certain organisms. So Neisseria gonorrhea has adhesins on the fimbriae or pili that adhere to cells lining the urethra and vagina of humans and cannot infect other hosts, for example. The virulence factors also include traits a pathogen uses to gain access to nutrients. So produce extracellular enzymes, and there's examples in the book of different enzymes that are produced in chapter 13. So they dissolve structural chemicals in the body to allow a further invasion and gaining access to nutrients. The pathogens have a lot to overcome due to our normal microbiota and our immune system. So if they can gain access into tissue where normal microbiota does not exist, they have a bunch of nutrients that they are able to access. So extracellular enzymes, toxin production, so there's exotoxins that are secreted by the pathogen in order to destroy host cells. There's different types: cytotoxins, neurotoxins, enterotoxins. Membrane damaging toxins such as hemolysins. This is one we're more familiar with since we use blood agar and we're looking for the lysis of the red blood cells. And then there's endotoxin, which is also referred to as lipid A, which is the lipid portion of gram-negative lipopolysaccharide. This is released when gram-negative cells naturally die, when they divide through binary fission, or are digested by phagocytic cells. Endotoxin can cause fever, inflammation, diarrhea, and shock. Virulence factors are also traits that a pathogen uses to escape detection and removal by the immune system. So there are antiphagocytic factors. The steps of phagocytosis are outlined in chapter 15. I usually go through it now to save some time in chapter 15. The steps of phagocytosis are chemotaxis, there's recognition and attachment, ingestion and formation of the phagosome, digestion, which is the fusion of the phagosome with the lysosome. The lysosome is a vesicle that contains chemicals and enzymes used to destroy the ingested antigen. And when those two vesicles form, the phagosome and the lysosome, forms the phagolysosome. At that point, the digestive enzymes have direct access to the antigen, so the bacteria or the virus, and the enzymes will destroy that pathogen. And then we get release of the digested antigen through exocytosis. There is a short supplemental video outlining the steps of phagocytosis that I would recommend you watch at this point. So I would pause this video and open up the phagocytosis video so you can better understand the antiphagocytic factors. So, with understanding the steps of phagocytosis, different antiphagocytic factors are needed for a pathogen to be successful. So capsule production is an antiphagocytic factor. Capsules are composed of chemicals found naturally in the body, so the immune cells don't recognize them as foreign. So antibodies cannot attach to the capsule material very easily. The capsules can be slippery, which makes them difficult for phagocytes to adhere. Some bacteria produce antiphagocytic chemicals, so the bacteria secretes a chemical in the phagosome and that into the phagocyte, and it prevents the fusion of the lysosome with the phagosome. So the pathogen is able to survive inside the phagocyte. The virulence factors are also traits a pathogen uses to avoid antibodies. Antibodies, again, are in Chapter 15. We'll cover them in more detail in Chapter 15. There's a supplemental video going over antibody structure and how they work as well, which I recommend watching after you go through this slide. Antibodies are Y-shaped proteins produced by activated B cells. Once activated, the B cells are called plasma cells. So the plasma cells secrete antibody. They're involved in the adaptive immune response, which is a specific response to antigens. So you'll get a specific antibody produced for a cold virus versus a specific antibody produced for strep throat, for example. Antibodies interact with antigens in a specific manner, so a lock and key type theory. They have a Fc stem that and a variable region. So the variable region is what attaches to the antigen. The Fc stem acts as a flag for phagocytes, allowing easier recognition and attachment. So again, I would recommend looking at the antibody overview video at this point. So in order for the pathogen to be successful, avoiding antibodies would be important. So since the antibodies are protein, some bacteria are able to secrete what are called proteases, so an enzyme that breaks down proteins, and these proteases break down antibodies. So some bacteria produce a protease that break down IgA in the body secretions that allow it to bind to adhesins on pili. Some bacteria rapidly turn over pili. So if the IgA binds to the pili, the bacteria basically breaks off the pili, since it can sense that something is attached to the pili, and produce new pili. So it's kind of like when you grab a iguana tail, for example. If the iguana feels threatened, they can break off the tail and allow the escape from the predator. Some bacteria can also do what's referred to as antigenic variation and actually genetically alter the type of pili made. So if the bacteria is being bombarded with antibody and these antibodies are attaching to the pili, that can cause a gene activation which allows the bacteria to produce a different protein on the tip of the pili. So not only can they do this antigenic variation, they can rapidly turn over the pili, and what this allows the bacteria to do is sustain itself longer in the human. So if it takes 2 weeks for an antibody to be produced for a specific pathogen, and the bacteria then changes the protein, that means the human has to go through another primary immune response. and that infection would persist for a longer period of time. FC receptors can also be produced by the bacteria. Like I mentioned in the antibody overview, the FC stem wants to be pointed out. This flags for phagocytosis. So some bacteria produce FC receptors, which bind the FC stem, and then the variable region is pointed out. At that point, the antibody is useless. Gonorrhea, for example, can produce chemicals that prevent the fusion of the lysosome with the phagosome and can secrete a protease that breaks down IgA in the mucus, and it can actually do antigenic variation of pili as well. If the bacteria can sustain itself long enough, avoid the immune system long enough, it can then leave the host through a portal of exit and find a new host. And again, it's human activities that are involved in the transmission of these diseases. Now, the last part of this is actually the information in Chapter 14 covering epidemiology. There's only a few notes here for Chapter 14. Epidemiology focuses on the effect of a pathogen in a population. So the study of where and when diseases occur and how they are transmitted in a population. So this field's important because the Earth is becoming more and more populated. Humans rely on more mass food production and distribution. We have increased travel to other countries. That leads to higher incidence, which is number of new cases of a disease in a population, and it leads to increased prevalence, so total number of cases in a population. So the occurrence of an infectious disease can be classified in terms of geographic distribution and frequency. So there's 5 terms here: sporadic, endemic, outbreak, epidemic, pandemic. So sporadic is a classification of random distributed diseases. So these would be very rare diseases. I separate sporadic and outbreak from each other in the textbook. Our textbook, these 2 terms are combined, so you'll want to know what's on this slide here. So an example would be the bubonic plague in the United States. So this is reported cases of human plague in the United States from 1970 to 2010. So numerous occurrences, but this is over 40 years, and most people don't realize that the bubonic plague still does exist. And you can see that there is a concentration in the southwest, usually in the western states here. And the bubonic plague is transmitted by a flea bite, which are usually associated with rodents. So sporadic would be a few cases randomly distributed, very rare. An outbreak implies a cluster of cases occurring during a specific, brief period of time, attacking a specific population. And typically you think of foodborne illnesses. So E. coli, 0157H7, measles, this would be another more and more common occurrence. Whooping cough, more and more common. Outbreaks. But whooping cough, for example, is actually endemic. But measles in 2019 here, we have 22 cases of measles originating from a visitor from Israel. So it's a specific population, people that came into contact with that person directly or indirectly, and they were not vaccinated, developed the disease. This figure here shows the occurrence of 77 people getting sick from salmonella outbreak from contaminated ground turkey. So this ground turkey was sent to specific locations, and a certain number of people got sick because they did not properly cook that ground turkey. Endemic is a native disease that prevails continuously. Usually the number of cases are pretty steady, with some spikes and declines. Endemics can lead to epidemics. Example here would be pertussis in the United States, which is endemic. This is the reported cases from 2000 to 2011. And overall, we're seeing a general increase in the trend, again, because people are not being vaccinated. Common cold, influenza would be 2 other examples of endemic diseases. Epidemic would be an a disease that is affecting an unusually large number of individuals in a population. So an epidemic may lead to a pandemic. An example here, H1N1 was epidemic in the United States in 2009, and then spread to more than 70 countries, so it was then pandemic. So endemic can lead to epidemics, and epidemics can lead to pandemics, which a pandemic is a disease affecting an increased proportion of the population over a wide geographic area, mainly worldwide then. So the best example would then be HIV/AIDS as a pandemic. So being able to stop the chain of infection is very important. So people going into healthcare have the ability to control nosocomial infections, which would be hospital-acquired infections. So disinfection, good housekeeping, bathing, sterile procedures, and hand washing would be the most important way to prevent or reduce nosocomial infections. So the CDC reports on average that healthcare workers wash their hands before interacting with patients only 40% of the time. So washing hands, changing gloves between patients is very important to reduce the spread of disease in the hospital. So factors that influence nosocomial infections, the presence of microorganisms in the hospital, immunocompromised people, people are at the hospital because they're sick, and then we have transmission of pathogens between staff and patients and among patients. So these three factors lead to nosocomial infections. Again, this is completely preventable. Well, not completely preventable, but hand washing alone is the #1 way to prevent these types of infections. This concludes the chapter 13-14 videos.